Hello, everyone. My name is Karthik Anantharaman. I am an assistant professor in the Department of Bacteriology at UW-Madison, where I've been here for about 4 years now. I don't know if you can hear this, but it's raining right now in Madison, and we're in the midst of a pretty heavy thunderstorm. So if you hear thunder, don't be worried. It's not coming from my office. It's coming from outdoors. So what I'll do is I'll just give a very brief introduction about my lab and the kind of research that we do before I get into Vibrant. So today's talk, I'm going to talk to you about a particular software that we have developed in our lab called Vibrant. Vibrant enables identification of viruses from different kinds of data, but primarily metagenomic and metatranscriptomic data. For the most part, we only work with metagenomic data as far as Vibrant is concerned. And if you're wondering why we chose the name Vibrant for our software, it's an acronym for virus identification by iterative annotation. We didn't plan on that; it just came about. So my lab, I run I lead a microbiome and virome laboratory here at UW-Madison. We really work at the interface of biogeochemistry, microbial and viral systems ecology, and computational biology. And there's two real questions really that motivate all of our work. One is we are trying to understand the role of microbes and viruses in driving sulfur metabolism. So if you followed our work, you'll realize that sulfur metabolism is a huge theme in our lab. And we are interested in sulfur metabolism from the perspective of biogeochemistry as well as human health and disease. So how does sulfur contribute to different human health and different diseases, let's say? And we're also trying to study viruses so that we can maybe even eventually modulate microbial communities. So we're a very diverse and interdisciplinary group. Now, I mentioned that we are interested in studying viruses and microbes and how they drive sulfur metabolism. So if you think of sulfur metabolism can be associated with organic sulfur or inorganic sulfur compounds. So if you're thinking about organic sulfur compounds, it can be amino acids like cysteine or methionine, or even taurine or taurocholic acids. There can be dissimilatory sulfur metabolism, and finally there can also be abiotic reactions. All of these are transformed into reduced sulfur compounds like H2S, which can really alter the states of entire ecosystems, including human systems, such as those that may be associated with diseases such as colorectal cancer, IBD, and so on. Now, what's interesting about all of these different processes is that it's different sets of microbes and viruses specifically that are associated with each process. And the association of viruses is something that we are very intrigued about, and it's a huge theme in our lab. When you look at these different processes, it also turns out that different systems are associated with different processes. So for example, in environmental systems, you're more likely to find dissimilatory sulfur metabolism than, let's say, organosulfur metabolism. Whereas in human and animal systems, for the most part, organosulfur metabolism dominates over dissimilatory sulfur metabolism. Now, in our lab, we also develop software tools to study microbiomes and viromes. So today I'm going to talk to you about Vibrant, but if you like any of our work, I encourage you to check out some of the other softwares that we've developed and put out, such as Propagate and Metabolic. So Propagate is a prophage activity estimator, whereas Metabolic can enable metabolic and biogeochemistry analyses in microbial genomes. We also have several other softwares in the work. So, stay tuned for work coming out of our lab. Now, as far as viruses are concerned, we are interested in three specific angles. First, we're interested in studying the diversity of viruses. So trying to ask a question such as how can we accurately characterize viruses and viral dark matter from microbiome data? We are interested in studying the distribution of viruses. So trying to understand viruses as predictive markers of ecosystem states or even a specific disease. And finally, an ecological angle. So trying to understand the roles of viruses in the environment. So today, I'm only going to talk to you about how we can accurately identify and characterize viruses. So Vibrant, the software developed by our lab, has been entirely developed and led by Christopher Kieft, a senior PhD student in our lab. Vibrant's the manuscript describing Vibrant was published in Microbiome in 2020, so I encourage you to check it out, and it's also available on GitHub. Now, when I thinking about the motivation for Vibrant, a long time back when we were actually discussing this, we were discussing the best way to identify viruses. And even back before we had automated tools to identify viral genomes from, let's say, metagenomic data, we relied on manual verification based on signatures of protein annotation. Essentially, if there are a viral genome, and you didn't tell us that this was a viral genome, just on basis of experience and looking at, say, manual verification, we could tell you that this was a viral genome. And this essentially served as an inspiration for Vibrant. So the idea that if we can look at viral signatures and determine that these are likely coming from viruses, can we benchmark this process? Can we make it reproducible as part of a software? And then release that so that others can also use it. So that was our motivation or inspiration for developing Vibrant. So we chose 3 different databases, KEGG, PFAM, and VOG, to annotate different protein families. And the idea being was that we can separate, or say we can distinguish between viruses and other microbes. The idea was, or the hypothesis was, that with regard to microbes would have more proteins that would be annotated better using KEGG or PFAM. Because these databases are richer in non-viral families, and VOG, which is a virus, or which stands for virus orthologous group, or is a database that's rich in viral proteins, this would help with identifying viruses. So this was our hypothesis. So on the basis of this idea, we developed a metric that we called VScores, and this is really the fundamental basis of Vibrant. So VScore is a metric of virus-like annotations. The idea again being that viruses have more virus-like annotations, and microbes, or say other non-viral organisms, have more microbe-like annotations. So VScore is a score that's assigned to each annotation or each protein family. VScores can go from 0 to 10. And it essentially describes how virus-like an annotation is. Okay, so it's not necessarily virus-specific, as in even a bacterial protein can be assigned a V-score, but the V-score for a bacterial protein will be very low, so it'll be almost 0, let's say, and a virus-like protein is going to have a V-score that's very high, so maybe like 10. So if you looked at a scaffold like this, an assembled genome scaffold like this, so you can see that a lot of these are viral proteins, so a lot of these would have very high V-scores approaching 10. So to give you an example of V-scores, if you had a portal protein, it would have a V-score of 10. On the other hand, you can also have a hypothetical protein with a V-score of 10. So what this is telling you is that we don't know the function of a specific protein, but this protein family is exclusively or almost exclusively found in viruses. And then if you had other proteins, then they're going to have much lower V-scores, as you see over here. So once we had V-scores, we could create a database of V-scores, and we could use that to train a machine learning algorithm that we can then use to differentiate between viruses and other kinds of genome fragments, or say genome fragments that come from other organisms. So what you're looking at over here is a plot of our machine learning algorithm once it's trained on data and then tested on other data. And what you're looking at are viral scaffolds in green, plasmids in red, and bacteria or archaea in blue. And what this is telling you is that our trained machine learning algorithm can differentiate between viral scaffolds as seen in green and other kinds of scaffolds. So how does Vibrant work? To give you a gist, Vibrant essentially identifies viral signatures. So those are the V-scores that I just described. It uses 3 different data sets or databases to differentiate viruses from microbes. And the fundamental idea is using a supervised machine learning model that also has some validated cutoffs. We think it has better efficiency than a lot of other programs. And a workflow, you can take any sort of input. We filter scaffolds at different points over here, depending on either the length or the number of open reading frames. So there are cutoffs that we work with. After this, we annotate the different proteins, and this eliminates about 75 to 85% of all non-viral scaffolds. This is then followed with our machine learning algorithm that eliminates the remaining non-viral scaffolds. And finally, we can conduct some virome analyses such as metabolism, genome completeness, and so on. So what can Vibrant actually do? Vibrant can identify lytic phages from genomic or metagenomic data. It can identify prophages, so phage that are integrated into microbial genomes. It can annotate genomes and phage genomes and determine genome quality. It can give you a sense of how phages are associated with certain metabolic pathways. So for example, it can identify auxiliary metabolic genes and the pathways that they might be associated with. And finally, it can help you visualize your data. So I'm not going to go into specific performance metrics really, but to sum up Vibrant's performance, Vibrant can be, we tested Vibrant on a variety of data sets from different sorts of environments, all the way from the human gut, wetlands, wastewater, soil, plants, all the way to marine environments and different kinds of marine environments. And Vibrant seems to perform very well. So, if you have an interesting ecosystem, you should certainly try and give it a shot. I mentioned that Vibrant can be used for determining the genome quality. So when it comes to genome quality, we implemented this, but I also encourage you to check out CheckV, because that can also help you determine the genome quality of a viral genome. As far as our own implementation goes, we define 4 thresholds for viral genome quality. So we call something a low quality draft if it's likely only the fragment of a viral genome, a medium quality if it's a significant portion, a high quality if it's the majority of a viral genome, and complete or circular, which is essentially the highest quality. So it's likely to be a complete viral genome. These quality metrics are estimated on the basis of presence of structural proteins, as well as proteins for replication, such as, let's say, DNA replication. Now, beyond this, Vibrant is also good at identifying and excising prophage genomes from bacterial genomes. So prophage is an integrated viral genome in a bacterial genome. And we use our VScore approach to accurately identify prophages and then excise them. This accuracy is pretty good, to the point that you can go in and find repeats within tens or let's say even hundreds of bases on the bacterial genome. What you're looking at over here is a bacterial genome shown here. And the colors over here are the phage genome. And I'm going to get at, say, how Vibrant enables identification of this bacterial genome. So it begins with cutting the bacterial genome at where we identify low V-score. So these in pink are low V-score. So these are definitely bacterial. After this, we identify viral-like annotations on either end. So on both ends, we cut the pink scores first. Now over here on this particular side, we've identified a viral-like annotation, which is the integrase. So we identified this as the threshold of a prophage. On this side, though, it's a little more complicated. The idea here is that these 4 proteins are hypothetical, whereas this particular protein has a viral-like annotation. So eventually we trim, so in this particular case, we have trimmed it to this particular location. These 4 proteins might comprise a part of the prophage, but we cannot be certain. And we typically try to work with higher confidence data. So that's the reason we choose this as the prophage. Now, Vibrant can also enable analysis of viral auxiliary metabolism between different environments, or even in your specific ecosystem. So this can be done at different resolutions, but just to give you a visual, just to visualize what you may be looking at. So for example, if you had one set of samples and let's say this was associated with the human gut, you would see something like this. So essentially what something like this was telling you is that in your data set, viruses have auxiliary metabolic genes that may be associated with carbohydrate metabolism, energy metabolism, and so on. Now, if you were to compare 2 different ecosystems or environments, in this case, let's say soil and the human gut, you can see that the profiles of auxiliary metabolism are very different. So essentially, the soil virome encodes very different auxiliary metabolic genes compared to the human gut microbiome. Now, I'll just mention one small technical detail associated with Vibrant. So Vibrant can be run on both mixed microbial metagenomes. So you're extracting viruses from mixed microbial metagenomes, or from viromes. So if you use the Virome flag, then it retains the pre-filters, but it loosens the criteria of neural networks, so specifically the machine learning implementation. And it can be used, it should be used more, rather for virome decontamination. And it seems to work really, really well when used with viromes. While we do expect that you have access to an independent server to run Vibrant, we have implemented it on the Cyver's discovery environment as well. So if you do not have access to a server, you can still run Vibrant online on the Cyver's environment, and I encourage you to check that out. I don't have a large summary, but hopefully I've convinced you that Vibrant is an excellent tool to characterize viruses from microbiome data. I encourage you to check that out. And if you have any feedback, questions, or thoughts, please feel free to get in touch with us. If you have liked Vibrant, I also encourage you to check out other tools from our software suite. And all of the work, we have a huge set of collaborators that are enabling all of our work. And I'd also like to thank our funders, without whose generous funding, none of the research would be possible. So specifically NIH, NSF, USDA, and JGI. Thank you all for listening. If you like our work, please consider joining us. We are looking for postdocs and grad students. We've got a very interesting set of projects. And with that, I'll take any questions.